initiate my video here for some reason. Let me see, okay. All right, much better. So Madam Moderator, thank you.
You're welcome.
I'm hoping you can see my screen and I'm audible.
Yes, you are audible and you can
okay.
see the screen.
So thank you again, Health-Edu for hosting and organizing this virtual workshop for journalists. And to the journalists, welcome all. And I hope that you will find this this workshop to be of journalistic benefit. Now, I have been asked to provide an overview of the current status of this fast shifting COVID-19 pandemic. I should begin by providing a disclaimer that I am not a clinician and neither am I a public health specialist. Unlike most of my colleagues who will be presenting to you throughout the course of this workshop, rather, I am a researcher. My area of expertise is in the area of the molecular biology of HIV infection here at Case Western Reserve University in Cleveland, Ohio. But I'm also a researcher who has a keen interest in this COVID-19 pandemic and also some research experience on the disease or on the infection, which I hope to have time towards the end of my talk to be able to share with you. So as we all know, SARS-CoV-2 is the pathogen that is responsible for COVID-19. And it is a zoonotic virus. That is a virus that has successfully crossed over from animal species into humans. And within the human population, it spread through the air several meters from one person to one another. And it enters the respiratory system initially, or the airways, and infects lung tissue. And you've heard that there are now these emerging variants of SARS-CoV-2 that have been shown to be more contagious or more infectious, but they are not any more harmful than the original strain. However, as I will present later in my talk, there are a few variants of concern that are not only more infectious, but also appear to be more harmful or more virulent. And this is a mysterious infection. And it's also, in terms of SARS-CoV-2 infection, and it's also a mysterious disease in terms of COVID-19 itself, because some people upon infection remain asymptomatic or develop only mild symptoms, while others develop severe COVID-19 and sometimes even succumb to the disease. So, and the underlying reasons for this wide-ranging variation in disease pathogenesis is still not clear. However, there have been risk factors that have been suggested for developing severe COVID-19, which include old age. So most of over 80% of COVID-19 deaths have been shown to occur in people over the age of 65, while more than 95% of COVID-19 deaths occur in people older than the age of 45. Secondly, people with pre-existing chronic health disorders that include cardiovascular disease obesity, diabetes, and hypertension. And thirdly, and these risk factors are suggested, the source being the Centers for Disease Control and Prevention here in the US. The third being issues like poverty and poor access to proper health care. However, in people that seem to appear healthy, that don't seem to have any apparent pre-existing health conditions and appear to develop severe COVID-19 and sometimes death, the question is there a genetic predisposition to developing severe COVID-19? And several studies have shown that indeed there is, and have identified regions of our human DNA, therefore genes whose polymorphisms increase the risk of developing severe disease. So these are based on genome-wide association studies that have identified genes or gene clusters whose polymorphisms increase the risk of severe COVID-19. However, a caveat or a limitation of these studies is that they were done regionally or globally spanning 19 countries in Western Europe and the US, but did not include any African country. Therefore, it remains to be seen whether these are the same genetic risk factors that might be also applicable in an African population. So there is a justifiable need to conduct similar genome-wide association studies with patients of African descent. Sorry about that. Okay. I'll pick up on this. Okay, my apologies for that interruption. My mouse dropped. Okay. So another point I would like to make in this presentation is that COVID-19 is not only a respiratory infection, but also a multi-organ systemic disease. So to the right here, I'm showing regions of the human body, and the solid lines point to organs or organ systems that are directly affected by the infection. Therefore, where the virus infects these organs and is able to replicate. The dotted lines represents organs or organ systems that are indirectly affected by the infection, where the virus may not necessarily infect cells, but is able to spread, or a consequence of an immune response to the viral infection is harmful to these organ systems. And to the left, I'm showing a schematic of the manner by which SARS-CoV-2 has been shown to enter cells. So it's an enveloped virus that expresses the protein spike on its surface that it uses to bind a receptor on the surface of cells called ACE2. And upon binding, proteolytic cleavage of spike by another protein or protease enzyme called TMPRSS2 is required for the virus to be able to fuse with the membrane of the cell and be able to enter the cell. So ACE2 is a receptor that is required for the entry of SARS-CoV-2 for the successful infection of cells by SARS-CoV-2. So once again, SARS-CoV-2 initially infects lung tissue, more specifically lung epithelial cells, and can cause substantial pulmonary disease, including pneumonia and acute respiratory distress. However, the virus has not only been detected in lung tissue, but also in these other organs, including the liver, kidney, heart, and blood samples in people who've succumbed to the disease. ACE2 is the receptor that's used for viral entry, and it's expressed not just in lung tissue, but also in other tissues and organs. And upon initially infecting the lungs, SARS-CoV-2 will enter the bloodstream and directly infect other organs that express ACE2. So the takeaway message from this slide is that that SARS-CoV-2 does not only infect cells of the lung, but it can also spread and infect cells of other organs that express ACE2. And the direct infection of multiple organs via ACE2 and the resulting hyperactive inflammatory immune response to the infection constitutes the basis for the disease, for severe COVID-19, for the systemic and multi-symptomatic manifestation of COVID-19. Then there's the issue of these long-term or long-haul effects of the disease that has been seen in some patients. If I'm not mistaken, in about a third of COVID-19 patients. So this is long after acute COVID-19, and also long after these patients are no longer positive for the virus based on a nasopharyngeal test, or even testing samples isolated from the respiratory tract. Okay, and these are some of the symptoms, some of the long-term symptoms that these COVID-19, these patients with post-acute COVID-19 syndrome face. And the question is, why is that? So once again, it's not the infection itself that's mysterious, but also the disease. Why do some patients, in addition to experiencing acute COVID-19, why do they end up also having these chronic effects? That's a question that still needs to be addressed. What also needs to be addressed is the clinical management of these chronic effects, these subacute to chronic effects of COVID-19. So getting to these variants, so regarded as the variants of concern. So how do they crop up? So SARS-CoV-2, like any other RNA virus, is prone to acquiring genetic mutations as it replicates and spreads. And variants with mutations that confer a replicative advantage, therefore increase infectivity or makes the pathogen more virulent, allows the virus to be selected for, or allows that those variant forms of the virus to be selected for, thereby ending up becoming the more dominant strains. And as of last month, 5 major variants have been identified globally. So the Alpha, which was initially identified in UK, the Beta variant, initially identified in South Africa, the Gamma, first reported in Brazil, the Delta variant, which is now becoming the more dominant global strain. Actually, over 80% of sequenced cases in the US have been reported to be of the Delta variant. This was first reported in India. And then there's this other variant that is now beginning to emerge. It's not really a variant of concern. It's still considered to be a variant of interest. But it's concerning because of the fact that it's not just more infectious, but appears to be more harmful than these other circulating forms of the virus. So to the right here, I'm showing some of the, a schematic of some of the non-circulating forms of COVID-2. And you will note that, so this is sort of like a schematic of the genetic structure of these variants. You will notice that the gene encoding the spike in all of these 3 variants seems to have the majority of these mutations. So therefore, the gene encoding spike is a hotspot for genetic recombination. And mutations in spike protein, particularly in its receptor binding domain. So the receptor binding domain of spike is the region of spike that binds to ACE2 in order to allow for entry into cells or infection of cells. So mutations in this region of spike that enhance its binding affinity to ACE2 will lead to COVID-2 variants that have higher infectivity. So to discuss more on these variants, so initially, as early as May of last year, there was a transition from the original strain, the original Chinese strain, into a mutant, this mutant here, that became the global dominant strain. And this mutation is on spike. It allows for a higher binding affinity to ACE2, and therefore for higher infectivity, and also therefore higher transmissibility. And it's been shown that patients with this mutation that have been infected with this variant of SARS-CoV-2 have a higher viral load due to the higher infectivity. Once again, it's more infectious than the original strain, but not more harmful. I've already mentioned that the Delta variant makes up over 80% of sequenced cases in the US and is soon expected to become the globally dominant form of COVID-2. This emerging Lambda variant, which was initially first reported in Peru, South America, is more infectious than the circulating forms. It also exhibits immune escape. So therefore, in viral neutralization assays, it's been shown that antibodies generated upon infection, a natural infection, or by vaccination are less effective in neutralizing the Lambda variant. What's interesting, so it's still a variant of concern. It's not really, sorry, it's still a variant of interest. It's not yet a variant of concern because it's just beginning to emerge. But what's interesting is that Lambda makes up 81% of sequenced COVID cases in Peru. So therefore it's the dominant strain in Peru. Coincidentally, Peru has the highest number of deaths due to COVID-19 of any other country. So in terms of deaths per 1 million people. And by comparison, the second highest number is in Hungary, which is half that is in Peru. So this Lambda variant, so these results suggest that the Lambda variant is not just more infectious and also able to exhibit a greater extent of immune escape, but is also appears to be more harmful. Okay. Then there's the question of these already approved vaccines. So one question is that I've been hearing, even from relatives and friends over in Rwanda and other parts of the world, you know, are these approved vaccines safe, and are they really efficacious? And secondly, can these vaccines offer immunity against variants of concern? So to the right here, boxed some of the approved vaccine types, which have shown good to excellent phase 3 clinical trial outcomes. So we have the mRNA-based vaccines, the DNA-based vaccines from Johnson & Johnson and AstraZeneca. And I should point out that these two vaccine platforms are the first of their kind ever to be used to vaccinate and protect against disease. Then there are the more traditional forms of the vaccine. So there's the Chinese attenuated, inactivated vaccine. And then there's this other vaccine that's not approved yet. It's a subunit protein vaccine from Novavax. But in a recent phase 3 clinical trial, has been shown to have a high efficacy over 90%, and also is quite effective even against a mixture of variants. So it's effective against COVID-2 variants. So the mRNA vaccines show greater efficacy. They appear to be more superior compared to the DNA and the inactivated vaccines. And there is emerging evidence that robust protective immunity conferred by mRNA vaccination can last for quite a while, for as long as 6 months to a year, but decreases with time. So this is based on recent data coming from the company Pfizer. And that when in mRNA vaccine-induced immunity is less protective against variants of concern. So it's still protective, but less protective compared to the original strain. And therefore, a recommendation has been made for a booster, and this recommendation is currently under consideration by these bodies here in the US, the CDC, the FDA, and the NIH. Immunization with the mRNA vaccines has been associated. So there's this concerning side effect. It's very rare with mRNA vaccination, but it's concerning. This has been found in particularly in young men below the age of 30. Immunization with the mRNA vaccines has been associated with a very low risk, but significant risk of developing mild myocarditis and pericarditis. But these can be easily treated with non-steroidal anti-inflammatory drugs. So to discuss more about these variants of concern, I know there'll be an entire presentation on these variants, but let me just touch a little bit on them. So there's a recent study, not published yet. It's still in preprint format. That's available out there that has shown that that these variant forms appear to show greater resistance by several-fold towards neutralization with antibody-containing sera isolated from convalescent or recovering patients or mRNA vaccinated individuals. So meaning that these variants can potentially escape the immunity that is conferred by mRNA vaccination. Secondly, the same study also compared effectiveness of vaccination with the DNA-based vaccine versus the mRNA-based vaccines in protecting against these variants. And they found that antibody-containing sera isolated from DNA-vaccinated individuals are significantly less effective at neutralizing all the major variants of concern as compared to sera obtained from mRNA-vaccinated individuals. So what about our children? I have 2 little children at home, one of whom will be resuming schooling in September. So is there a reason for concern that our children still remain unvaccinated? There are ongoing clinical trials to determine whether the already approved vaccines are safe and efficacious in children. But is there a reason for concern that our children remain unvaccinated? And the answer is really no. Children seem to have a much reduced susceptibility to developing severe COVID-19. So they've thus far, and I'm saying thus far here in bold, because we don't know what the future holds in terms of these variants emerging, but thus far, they've been largely protected from developing severe COVID-19 and related pneumonia. And as of the 15th of this month, and just, this is just an example, about 14% of all confirmed cases in the US were children, while only 1 to 4% of all hospitalized cases were children. And therefore, identifying the basis for the much reduced susceptibility to COVID-19 will be crucial to understanding disease pathogenesis, but also providing clues for effective treatment. So some of the proposed reasons as to why children have a much reduced susceptibility to severe COVID-19, and I'm showing it in this schematic here on the right, is one, that these children, because they have a greater frequency of infection with viruses that cause the common cold, including other coronaviruses, they do have pre-existing reactive immunity that when they encounter an infection with SARS-CoV-2, they are well protected. Secondly, they have a reduced expression of receptors like ACE2 on their alveolar epithelial cells of lung tissue so that the virus has a limited chance of infecting their lung tissue and spreading to other parts of the body. And thirdly, in general, they have a stronger immune response to an infection. And lastly, and this is very important, so unlike the adults, children tend to have a lower hyperactive or an overreactive immune inflammatory response to infections. Okay, so touching more on the vaccines. So this, here I'm showing pictures of an event that occurred last week here in the US. This was the NBA finals. This was last Tuesday, and I'm showing a large crowd of people in Milwaukee, Wisconsin. Over 65,000 people attended this event. This is outside the arena, the basketball arena, and about 17,000 people were inside the arena. Most of them were not wearing masks, and so this would be considered to be potentially a super spreader event, right? But wait, most of these people were vaccinated. And so my point here being, these approved vaccines can allow an event like this. We haven't had in the news that there's a consequence of this event happening last week, because most of these people are vaccinated. So I believe in the anecdotal evidence that is out there that the already approved vaccines are indeed highly protective against symptomatic disease. However, older adults and immunocompromised individuals, they seem to be, or they're reported to be at a higher risk of a breakthrough infection. That is an infection occurring in a person who's already been fully vaccinated. But in most individuals that become infected, so those that are vaccinated and have a breakthrough infection, even in these what would be considered to be higher risk categories of people, the illness is much less severe and there's a reduced need for hospitalization. I would also like to make the point that a major reason for breakthrough infection, even among seemingly healthy individuals, is the inability of a population to achieve herd immunity. Okay, so for instance, here in the US, there's an oversupply of vaccines. And so if you want to get vaccinated, if you're eligible and want to get vaccinated, you will get vaccinated. But there is a significant, a sizable percentage of the population that has declined the vaccination or the vaccines, about 40% of adults. And therefore, even in the US, we are unable to achieve herd immunity for COVID-2 infection. So therefore, overcoming, and there's an irony here that we have an oversupply and yet people are declining the vaccines, but back in the developing world, there's a dire shortage of approved vaccines. So overcoming this dire shortage of approved vaccines and the elimination of hesitancy or refusal to get vaccinated due to misinformation or political motives over here in the US will be crucial to achieving herd immunity. So What's the progress with the development of rapid and more affordable testing? So the goal here, especially in the developing world, is to test as many people in a short time and affordably while maintaining accuracy in viral detection. So the RT-PCR test still remains the gold standard for testing. So this is testing of nasal and oropharyngeal samples, but it is expensive and comes with a slow turnaround of about a day, one to 2 days. And then these other tests that are considered to be more rapid, antigen-based and serological testing, they are not reliably accurate. And part of the reason being, there is substantial error that comes about in picking up false positives. Therefore, cross-reactivity with viral antigens that are similar to COVID-2 spikes, or in the case of serological testing, antibodies elicited against these antigens. So I would like to point out this study here that was published by a group based in Rwanda. It's really a commendable effort where they've developed a strategy to test as many people using the gold standard RT-PCR, okay, but in an affordable manner. So see here I show their proposed model, which they refer to it as a hypercube model for the identification of infected individuals in a large group of people after a few rounds of pool testing. So they can even identify if there's a one positive case in 100, they are able to identify that. Okay.
Hello.
Yes.
I'm sorry
I'm almost wrapping up here.
Right. I think you can use 5 minutes to wrap up because you're running out of time.
Yeah, I should be done within 5 minutes. Sorry about that.
Thank you.
Thank you. All right. And they also show that in a pooled test of samples, they can accurately identify positive specimens by RT-PCR. Okay, so this was just a proof of concept. This was diluting a positive sample 100-fold, and they are able to show that they can show that it turns out to be positive in an RT-PCR test, well below the threshold count of 40 cycles. And they are able also to show a correlation, an inverse correlation between the prevalence of a viral infection and group size. So they're able to identify optimal group sizes based on the prevalence of viral infection. So what about treatment? So treatment with repurposed drugs and monoclonal antibodies. So make the point that there isn't any clinically proven strategy yet for COVID-19 treatment other than supportive care with repurposed drugs, oxygen therapy, and monoclonal antibodies. And this depends on disease severity. So there is antiviral drugs, the most effective of which seems to be remdesivir. There is the anti-malarial drug hydroxychloroquine and immunosuppressive drugs that are used for supportive care. And in addition to that, early intervention with monoclonal antibody treatment can reduce the risk of severe illness and prolonged hospitalization, especially for people in the high-risk categories. And there's recent news that, good news that these monoclonal antibody therapies have now been approved for manufacture by India, which makes them, which should make them more affordable. So I'll discuss with you in the next or so the research that I've been doing here in case as it relates to COVID-19. So this has involved the expression and purification of these structural proteins of COVID-2 in an effort to screen sera convalescent sera or sera from vaccinated individuals to identify effective neutralizing antibodies. I should point out that most neutralizing antibodies that are generated either by vaccination or a natural infection are targeted towards the S1 region of spike, the region that binds to ACE2, that has the receptor binding domain. So successful ectopic expression and isolation of these structural proteins has allowed for screening of convalescent sera to identify antibodies that cross-react well with each of these structural proteins, and also antibodies that can be isolated and further assessed for their potential to neutralize the viral infection. So here I'm showing a procedure that we call Western blotting to demonstrate that we've successfully expressed these recombinant forms of the structural proteins in a lung epithelial cell line. And by microscopic imaging, we are able to identify how these proteins are distributed in cells. And I'll be able, in the next few slides, I'll be presenting these microscopic imaging results to you. So this is imaging of the spike protein in lung epithelial cells. So in green is the spike protein, in blue is the nucleus. And you can see that the spike protein is distributed along the plasma membrane, which is marked here in red. These are cells that express both spike and the nucleocapsid, and you can see their differential location patterns. So in green is spike, it's localized or distributed along the membrane, while nucleocapsid is within what we call the cytoplasm. And then what about the M protein, the membrane protein? So the membrane protein appears to be intracellular, so expressed within the cell. And this there's some reason for concern with this, because as I've shown you here on this slide, the membrane protein is expressed on the surface of the virus, but in cells appears not to be on the membrane, but within the cell itself. And so there's a reason of concern as to whether membrane protein might be able to elicit effective neutralizing antibodies, and it's something that we're looking further into. So, and as the pandemic evolves, so must the research. So for instance, with regards to the own research we're doing, identifying antibodies that are effective against COVID-2, now we have to consider variants. So for instance, instead of cloning the original and expressing the original spike, now beginning to express all these variant forms of spike, doing the same experiments. Upon expression, purifying or exposing cells that express these variant forms to sera from convalescent or vaccinated individuals in an effort to identify monoclonal antibodies that cross-react with the variant forms, then perform the viral neutralization assays to identify antibodies that with potential for treatment. And so with that, I'll close by summarizing that COVID-19 is a multi-organ systemic disease that initially disrupts lung function, but can spread to other organs that express ACE2, which is the surface receptor used for viral entry. That direct infection of multiple cell types via ACE2 and the resulting hyperactive inflammatory immune response constitutes the basis for severe COVID-19. That there's a need to conduct these genome-wide association studies to assess genetic risk factors in patients of African descent. That the Lambda variant is not only, which is now beginning to emerge, is not only more highly infectious, but also exhibits substantial immune escape and appears to be more virulent or more harmful. And identifying the basis for the much reduced susceptibility to develop COVID-19 in children will in turn be crucial to understanding disease pathogenesis and providing clues for effective treatment. And lastly, in the absence of effective treatment strategies for COVID-19, drug treatment strategies, affordable monoclonal antibody may be the best bet for reducing risk of severe illness and prolonged hospitalization. I thank you all for your attention, and sorry that I went over my time. And I thank the moderator for being patient with me on that. And I'll be glad to take any questions if there's any chance to take any questions. Alternatively, I can also accommodate questions on the chat feature. Thank you all.
Thank you so much, Dr. Uri. I think the questions, if there are any, I can see some in the comment section. They will be answered in the chat box or at the end of the session. For now, because of time, let's welcome our next presenter, who is going to be Dr. Felix, to give us a session on what he has prepared. Dr. Felix is a medical doctor and specialized training in disease diagnosis from the University of Rwanda. He is currently also a doctor of pathology at the University of Rwanda School of Medicine. You're welcome, Doctor. Hello?
Thank you. Hello, thank you. I was under mute. Now I think you are hearing me. Hello, are you hearing me?
Yes, I'm hearing you.
Okay, thank you so much for this great opportunity. As they introduced me, my name is Felix Manirakiza. I'm a medical doctor. And I'm a lecturer at the University of Rwanda in the School of Medicine. Now I'm doing my PhD studies in Japan. I have seen through the chat some people are claiming the use of some Kinyarwanda. So I don't know if that is, if you want for some terms that to be used, to be explained using Kinyarwanda words. Do you want that?
I think we can use that for some terms that will need explanation from you.
Yeah. Okay, thank you so much.
Sorry, Doctor, just a quick reminder, we're using 20 minutes for the presentation and 5 minutes for question and answers. Thank you.
You are getting my presentation. You are able to see it?
Not yet.
Not yet. Now you can see my presentation?
Yes, you can see it.
Okay, I got to talk about introduction and background of COVID-19. First, I want to remind you that COVID-19, it is a coronavirus disease, an illness that affects human, as Dr. Uri has explained, and is caused by coronavirus 2. I emphasize for this number 2 because there have been misunderstanding when people have been saying that coronavirus was known before this pandemic. There have been other coronavirus which did not which did not cause COVID-19. So those are the coronaviruses which have been there since a long time. And as we'll see, some disease caused by coronavirus other than coronavirus type 2 caused severe acute respiratory syndrome in 2003. So this disease has been declared a pandemic by the World Health Organization and is having severe effect on both individual lives and economies around the world. Here I put these words: pandemic, that is different from epidemic, that is different from endemic and outbreak. Maybe because of short of time, as we have the presentation, we will read them for more explanation. So epidemic, a disease in a large number of people, while pandemic, it is an epidemic, but we've spread in multiple countries and continents. Endemic, when the disease is like localized for particular people or particular countries. For example, we say malaria. Malaria is endemic, for example, in Africa, but we not find malaria. For example, in Japan, we have rare cases of malaria, or not at all cases of malaria. So malaria, it is endemic in tropical areas. This disease was called COVID-19 because the first cases occurred in 2019. And here in the title, I write COVID-19 / severe acute respiratory syndrome, COV2 for coronavirus type 2. Because as medical practitioner, someone may say this person have COVID-19, the other one may say this person have COVID-2. So these are terms or terminology that can be used interchangeably. So I put this to make that clarification. So the first outbreak of severe acute respiratory syndrome caused by COVID, other than COVID number 2, occurred in February 2003 in Asian countries. At that time, as this COVID is different from the COVID we are now having, only 8,098 people worldwide become sick, and only 774 died. So this was a severe acute respiratory syndrome that was caused by COVID, which is different. That was caused by coronavirus, that is different from the coronavirus type 2. These viruses has been called corona because, as you are seeing on the picture, it has a clone-like. When you look at this virus under the microscope, you will see the image like this with some clone. So it is there, it is getting its name. The viruses are named based on the genetic structure, and this is done to facilitate the development of diagnostic tests, vaccine, and medicines. This name of coronavirus type 2 was chosen because this virus was genetically related to the coronaviruses responsible for the severe acute respiratory syndrome outbreak of 2003. But as I have been explaining, even if these two viruses are related, but they are different. So they have a relationship, but they are different. Coronavirus that causes COVID-19, it is an RNA viruses. So I put this in red because, for example, in our country, most of people, sometimes they say we will go to do DNA tests. Someone has gone to do DNA tests. So these DNA and RNA, they are somehow similar, but they are different. DNA and RNA are the genetic material, are the building material of our body, of any living, either plants, animals, viruses, and microorganisms. So some of living organisms, their genetic material is made by DNA, while for others, the genetic material is made by ARN. And especially in viruses is where we have some viruses which are made by DNA, while others are made by ARN. And as here I explained, there is a difference between DNA and RNA, but maybe these are on the level of those who do research in medicine. As some history of COVID-19, we have to remind you that the first cases of COVID-19, or the first patient to have COVID-19, were seen in China, in Wuhan municipal health area, where a cluster of cases of pneumonia occurred. So when you are saying pneumonia is what in Rwanda we call umusonga. So there were people who were having signs and symptoms that were showing that they have problem with breathing, shortness of breath. So those people were first found in Wuhan, China. On the 4th January 2020, World Health Organization reported that there was a cluster of pneumonia cases, but until that time, there was no death. So on the 5th January, World Health Organization published its first disease outbreak news. So we can say that it is since 5th January 2020 when this disease was known as an outbreak, because numbers of cases were increasing so rapidly. On 10th January, World Health Organization issued a comprehensive package of technical guidance online with advice to all countries on how to detect, to test, and to manage potential cases of COVID-19. So all these are some history. So because of short of time, you will have these notes, and then you will have a look at them. In Rwanda, we can say that the first case of COVID was recorded on the 14th March 2020. And the first death occurred on 1st of May 2020. And as now you know, and these days you have around 10 cases of death every day. So what are the possible origin of COVID-19? What I can say about this question is that the possible origin until now is not, we don't have any conclusion, no conclusion yet about the origin of the COVID-19. There are some theories or hypotheses. Some are saying that COVID, it has a natural origin, while others, the other people are saying maybe COVID-19 comes from a leak of viral microorganism from a laboratory in China, while other people are saying that there have been viral engineering. So with genetic engineering, people have made viruses that are causing COVID. So what I can conclude to the information that we have until today is that is better we wait for the result of scientific investigation, because investigations are still going on. So, and the current information is that some is that some facts or some data that are towards a natural origin, but also people, they are still thinking about a possible leak, because if this is a case, so people can take some measures to prevent a similar thing to happen in the future. So I want to talk a little bit about signs and symptoms of COVID-19. First, I have to make some clarification. When we are saying signs and symptoms, these are two different things. So signs of a disease, it is an objective characteristic of a disease, while a symptom, it is a subjective. So cyangwa se mu yandi magambo nkanga nabyo reka mbisobanure gato mu Kinyarwanda. Iyo tuvuga ngo sign of a disease, urugero natanga, iyo umuntu ashaka kubwira ngo umutwe uri kundya, ntabwo ushobora kumenya niba koko arwaye umutwe cyangwa se atawurwaye, kuko umuntu ashobora kuvuga ati ndi kuribwa umutwe akubeshya. So that is subjective. Araza kubwira ndwaye umutwe ariko ntabwo wowe ubimenye. So that is subjective. So we call that a symptom. But umuntu naza yavunitse akaguru habyimbye bigaragara, nawe ushobora kwibona n'amaso, because we are able to see that he has that swelling, we call that a sign. So a sign, it is objective. Anybody, other people can see, can confirm that. While symptoms, just you will believe in what people are telling you. If they said I have pain, you will believe that they have pain. But you cannot know exactly if they have pain or not. So these are some signs and symptoms of COVID, and some have been explained by Dr. Uri. I go again talk a little bit about COVID variants. When Dr. Uri was presenting, he mostly talked about a variant of concern. So when you have the variant of COVID are grouped. Now, until now, we have two groups, because the third group, as we'll see in the next slide, in the third group, we don't have any case until now, any variant. But for other viruses, when we are classifying them, we have viruses, we have variant of concern, variant of interest, and variant of high consequences. And we make differences for this variant or for this group based on some criteria. So we call it a variant of concern when we have evidence of an increase in transmissibility. So when there is, when the possibility to transmit the disease from one person to another is high. And when you have, there are more severe disease, when the disease, the virus is causing is more severe. So we'll say the disease is more severe, for example, when many people will go to hospital for being hospitalized, or when you have many people who are dying from the disease. We'll call a variant of interest if there is some specific genetic markers that have been associated with changes to receptors binding, if there is reduced neutralization by antibodies. So for example, when the immunity has a low capacity to neutralize, to contain the viruses, so in that case, that variant will be classified among a variant of interest. We'll call a variant of high consequence if there is clear evidence that prevention measures or medical countermeasures have significantly reduced effectiveness relative to previous circulating variant. For example, when referring to other disease that you have been knowing, I can say, for example, in the case of Ebola, even if Ebola is different from COVID, Ebola should be classified in a virus of high consequences. As Dr. Uri explained, until now we have five variants. So he explained when in his presentation, he told you he told about 5 variants, but maybe I got to talk about more than 5, because in my classification, I did classification according to these 3 classes: variant of concern, variant of interest, and the variant of high consequences. So most of the time now, most people are talking about variant of concern, which are alpha, beta, gamma, delta, while the lambda is still classified in variant of interests. And this is what Dr. Uri has explained. Yes, it is. Now we have more interest on variant lambda because it's causing more death and so on. Maybe in coming days with more facts, it may be classified among variant of concern. But until now, up to date, is classified among variant of interest. So as I said, until now, we don't have any variant registered in category of variant of high consequence in variant of COVID-19. So why or how do we have COVID-19 variants? Ni gute cyangwa se ni mu buhe buryo nyine tubona variant cyangwa se iyo bavuga mutations? Variant emerge from mutation. So I want to explain mutation using some simple terminology or Kinyarwanda words. A mutation, reka ntange urugero mu Kinyarwanda. I give an example in Kinyarwanda, just to try to give something that you can understand easily. Ndatekereza ko abenshi muri mwe mwaba mwaracurishije nk'urufunguzo, hari igihe umuntu akenera kugira urufunguzo rwa kabiri. Urufunguzo rwa kabiri, iyo wari ufite urufunguzo original ukajya gucurisha urufunguzo rwa kabiri, rwa rufunguzo hari igihe rimwe na rimwe ruba rudafungura neza nk'urusanzwe. Nuvuga ngo haba hariho akantu gato cyane, haba hariho a small change, a small differences from the original key you are having. So the mutation is like that. A mutation, it is a small change. It is a small change that occur in genetic material, and sometimes it may be harmful, sometimes it may be harmless. So when you got that change, you may have problem if you had that change in genetic material. So these changes occur during replication or division or multiplication of viruses. Nkuko natanze urugero ku mfunguzo, uko uzajya gucurisha urufunguzo, uyu munsi urashobora kuguha rufungura exactly nka rwa rundi ruri original, ariko nujya gukoresha gucurisha urundi rufunguzo uhereye nanone ku rwacuzwe mbere, at the end ushobora kuzagera ubwo bazabagwa none n'urufunguzo rudafungura, cyangwa se bakabagura urufunguzo rufungura imiryango 15. So that is how mutations are also occurring in viruses and in other living organisms. Here, using technical terms, normally a mutation, it is a permanent error in a DNA or RNA, a permanent error. Normally, when our cells are dividing, or when viral genetic material are dividing, are making multiplication, there are some errors which are being occurring. But in normal condition, those errors are being corrected. So the problem with viruses is that viruses are dividing very, very, very quickly. As you see, I put it in I highlighted this word. So viruses are dividing too quickly. So if they are dividing too quickly, there will not be enough time to repair those errors. Ngira ngo hano murabyumva neza. Ni ukuvuga ngo no mu muntu, no mu buzima bw'umuntu, iyo serire z'umubiri we ziri kwidiviza, iyo DNA material yacu iri kwidiviza, iri gukora division, iri gukora multiplication, hagenda habamo udukosa. Ariko hariho mechanism ubusanzwe muri normal condition, mu bihe bisanzwe, there are some mechanism to repair those problems. So problems are occurring, but those problems are corrected. So iyo rero ufite umwanya uhagije, all problems will be corrected. Ariko niba atariho umwanya uhagije, some problems will not be corrected. So if those problems are not corrected and they are maintained and they become permanent, they will be called limitation, and they will be causing problem in future. And I think that we'll get more explanation about COVID variant in coming in the presentation that will be offered on Friday. The other thing that we can say about COVID-19 is about the vaccines. Dr. Uri also have talked about this. So as you know, we have a lot of information about COVID-19. So now I think he talked about only free vaccine, but we, as you know, journalism, most of the time we have information from different sources. What I have to add there is that we also have the vaccine that is made from Russia. Some countries are skeptic to adopt this vaccine from Russia, but this vaccine is being used in around 70 countries. And the current evidence is showing that this vaccine is safe at some level, like other vaccine we are using in the USA. So as take-home message, we can summarize that we have to know that COVID-19, it is a pandemic because all continents or countries are affected. I emphasized that it is caused by coronavirus type 2 because I want to make the difference between other coronavirus, which caused other disease were different from COVID-19, with this coronavirus which make the disease, which is called COVID-19. This disease has and will cause very important economic losses. As you know, we've locked down, with suspension of some activities. So this disease will cause some important economic losses. Signs and symptoms vary from one person to another person. The other thing, or in another example that I can use to explain this, reka nawe nyitange mu Kinyarwanda. Urugero, ngewe iyo ndi gusobanurira abanyeshuri ibijyanye na signs and symptoms, no kuba abantu bamwe barwara, umwe akaremba cyane undi ntarembe cyane. Sometimes this is caused by our genetic material. Kuba se umubiri wacu usanga mu bundi burwayi, nuva niba umuntu asanzwe afite ubundi burwayi, ubudahangarwa bw'umubiri we ari bukeya, abasirikare ari bakeya, uwo muntu ashobora kugira ibimenyetso byinshi kurenza umuntu ufite amagara mazima nk'uko bivuga mu Kinyarwanda. The other thing is genetic material. Our genetic material, our DNA is different. So some people are resistant while others are less resistant. Urugero ntanga iyo ndi kubisobanura abanyeshuri, ndababwira ngo abantu batanu baba mu rugo rumwe, baba wenda niba barara mu nzitiramubu kandi bose bayiraramo, hari igihe usanga hari umuntu umwe uhora arwaye malaria, abandi ntayo barwaye, kandi abo bantu basangira ibintu bimwe, bavuka mu muryango umwe. Cyangwa se nuvuga ngo urwo n'urugero rufatika cyane rutwereka how we are different. Tugendere kuri malaria, twese tuzi why. Ushobora kwibaza uti nukwerekera umuntu umwe mu rugo abantu baba hamwe, basangirira hamwe, imibu iba iri kwarira rimwe, cyangwa se bose barara mu nzitiramubu, cyangwa se nibayiraremo, umuntu umwe agahora arwara. So because our immunity, our body is somehow different in regards of how we how we fight against the diseases. So as I explained, until now, we have 4 COVID variant of concern, but with other variant, some in interest category, including Lambda. And the last information is that you have free COVID vaccine approved for use in United States and in other countries. So these are approved in the United States and are more acceptable in many countries, while as I explained, the other one vaccine that is developed from Russia is most used in Russia, but other countries also have started adopting this vaccine. So thank you so much for your kind attention. Your questions are welcome.
Thank you so much, Dr. Felix. Hello?
Hello.
Yes, thank you so much, Dr. Felix. The session has been good and great explanations, Mr. Rwanda. If there are any questions, I think we'll also use the same method we used with Dr. Uri in the chat box below. Maybe at the end of the session, towards the end of the session, we'll have a one-on-one where we'll have questions from the participants and let them speak and ask questions. For now, I think you can answer some of the questions in the chat box below. Thank you so much.
Thank you so much.
I would also like to thank Dr. Uri and Dr. Daisy in the group chat who are replying to most of the questions in the chat box below. I'd also like to thank the participants, the journalists, for the participation. Everything is going on well. Thank you so much. I would like to welcome our third presenter, who is Dr. Simba. Dr. Simba is a Rwandan medical doctor and global health researcher who has a PhD from Nagasaki University with the experience of more than 10 years in the health sector, as well as on the international health scene. You're welcome, Dr. Simba. Before Dr. Simba starts, I would like to remind the participants, please change your names, your device names to your real names. I can still see ARJ07. Galaxy Infinix, please change your names to your real names. Also, if you're not able to join the session because maybe the number of participants is full, you can follow us on YouTube. We are live Health-Edu YouTube channel. These courses and the presentations will be on the Health-Edu platform, so you can be able to get them when you log onto the Health-Edu platform. Thank you so much. Welcome, Dr. Simba. Hello, Dr. Simba.
Okay, good. I was trying to unmute myself, so now I think I can.
Okay.
So ndaza kubasaba muze kunyihanganira sinza gushyiraho video umwanya munini ariko najya mbasuhuze kandi komeza no kwihanganisha abanyarwanda mu bihe bikomeye turimo bya COVID-19 na za lockdown, si mu Rwanda honyine n'isi yose ariko iwacu mu Rwanda birumvikana n'umwihariko. Nk'uko babivuze rero nitwa Akintije Simba Kariope, nkaba ndi umuganga, nkaba ndi umushakashatsi muri Nagasaki University, especially muri Nagasaki Institute of Tropical Medicine. So ndaza ro guca muri make kuri pathophysiology ya COVID-19, sinza kuyitindamo cyane kubera ko umwanya waturangiranye, ndagerageza gusummarizinga iby'ingenzi mbona bikenewe cyane. And I will mostly use Kinyarwanda, so I think it's not forbidden. Kugira ngo maybe I feel more comfortable with that.
Yes,
so please welcome to my presentation. reka ntangire n'icyo twita disclaimer. Mbanze nshimire abambanjirije Dr. Uri and then Dr. Felix. They have really elaborated much more on COVID-19. Ariko uko ngiye kuvuga ndabanza ntange disclaimer ko I'm not really an expert of COVID-19, even though no one now can really claim he's an expert, he or she's an expert. And also I'm not a virologist. Virologist ni bari abigiye by'amavirus bakabyinjira cyane. I'm just a medical doctor and a medical scientist, of course, with an interest in infection research, but hands-on global health, especially maternal and child health. Those are my area of expertise. And then public health diplomacy and infectious disease intelligence aspire. So those are my really point I want to mention before I start. So COVID-19, bamwe bayitiranya, bamwe bati COVID-19 ni indwara, abandi COVID-19 ni virusi. So harimo ibyo abambanjije bagiye bavuga cyane, banagarukaho. Ariko ubundi COVID-19 ni indwara, ni izina ryahawe COVID-19, kimwe nuko na malaria tuyita malaria, hakaza n'izindi dushobora kwita amazina za HIV, iziterwa na HIV tukazita sida. COVID-19 rero nayo ni indwara. Amazina rero ya virusi ubwayo agiye atandukanye, ariko cyane cyane iyo tureba hano iyo bita SARS-CoV-2, Dr. Felix yagarutseho cyane, yabajwe no kwitwa 2019 nCoV, hari na COVID-19. So ni amazina bagiye bayiha agiye atandukanye, ariko the current one which is really the more concerning ni SARS-CoV-2, kandi binahereye ku mateka hari zagiye zibanza COVID-1, MERS, CoV, hari zagiye zibanza, abantu bashobora no kuba banabisoma online. Actually most of the information now on this kind of things ziraboneka online. SARS rero nkuko nabivugaga, SARS-CoV-2 ni severe acute respiratory syndrome coronavirus 2 and ni infective agent behind COVID-19, kandi tubizi neza ko yirihuta cyane mu gukwirakwira hirya no hino, kandi morbidité na mortalité, cyangwa se tuvuze morbidité na mortalité ni ibintu bibiri, donc uko itera indwara nuko yica biri hejuru cyane. Kugeza uyu munsi ku itariki 28 z'ukwa karindwi ku isi hose dufite 197,000,000 z'abantu bamaze kwandura babaye confirmed, icyo twita confirmed cases. Hakaba harimo miliyoni hafi 4,200,000 by'impfu zimaze kugaragara. Aho ni ku isi hose. Twajya mu Rwanda iwacu ngira ngo situation muranayindusha kurusha, tumaze kugira ka ziri confirmed 36 n'impfu z'abantu bamaze guhitanwa na COVID bageze kuri 471. Iyi ni imibare y'uyu munsi ya WHO OMS. Turebye rero mu Buyapani bari banasabye ko nshobora gutanga na some hint about situation on Japan. Japan kwera ko ari nihomba, sinena bivuze ariko nihomba. Kugeza uyu munsi dufite 892 by'amakesi, ni cumulative cases ziri confirmed. Actually Ubuyapani ni igihugu kiri mu bihugu byakomeje kugerageza kulimita umubare w'abantu bandura cyangwa se kurwana n'iyi ndwara, kuko kuri miliyoni hafi 140 z'abaturage bafite 800,000, ntabwo turagera kuri miliyoni y'amaka. Gusa muri no minsi kubera Olympics turabona kazi kuzamuka, kuko murebye nka hano kuri sinzi niba nshobora kubona uko nkoresha pointer. I'm not really an expert in these things. Anyway, ndabona ntarimo mbibona, ariko uyu munsi twagize kugeza ku isaha ndimo nkora presentation tumaze kugira ka 9,000. Ni ukuvuga ngo ibi ni ibintu bitari bisanzwe, twaga tugira ka 3,000 ku munsi, 2,000, ariko ejo byari 7,000, uyu munsi byabaye 9,000. Ni ka zero ziri hejuru cyane ku buryo tubona harimo ikibazo. Ikindi nuko death, abapfa bahagaze ku 15 kugeza uyu munsi. Uyu munsi ubwo nari nateguraga presentation ko tumaze kugira abantu 6 baramaze kwitaba Imana. Twibaza ko nabyo ni ibintu bidasanzwe, kuko ubundi ndabona ndabigarukaho, ntabwo indwara yajyaga itesha cyane hano mu Buyapani, nubwo nyine ibibazo bitabura kubera mesure za prevention cyangwa se preventive measures kubumva igifaransa, byatumaga imibare ikomeza kuba hasi, ariko ubu birimo birazamuka. Iyo turebye kuri chat iri hirya iburyo, ndibaza ko ari iburyo, ku maka urebye ku myaka abenshi n'abantu b'urubyiruko, imyaka 20 kugera kuri 30, cyangwa se kugera kuri 20 kugera kuri 40 muri izo kategori. Abasaza n'ubwo hano igihugu gifite abasaza benshi, ariko ntabwo baraba benshi bafite ubu burwayi. Ku kijyanye na vaccination mu Buyapani, uyu munsi tumaze kugera abaturage miliyoni 81, hamaze gutangwa doze miliyoni 81. Ni ukuvuga ngo abaturage bari fully vaccinated ni miliyoni 34, bagize rero 26.5% by'abaturage b'Ubuyapani. Nk'uko mwabibona ku ifoto, urebye hano hari preventive measures of course, ariko most of the time ubuzima burakomeza, haza ibyo bita state of emergency, abantu bakagabanya ingendo, bakagabanya guhura n'abantu icyarimwe, ariko ibisanzwe akazi birakomeza na economic akomeza kuzamuka. Bafite uko badiringa nabyo, kubera ko impamvu nimwe bigeze Dr. Felix yabigarutseho, bigeze bagira ibyorezo n'ubundi bikomeye nk'iri ngibi bya SARS zabateye, za influenza n'izo za MERS-CoV, ni indwara na za tuberculosis, ni indwara zabahaye ikintu cya resilience. Nanibaza ko no mu Rwanda cyangwa se muri Afurika muri rusange ni ibintu tutari tumenyereye, ariko nkeka ko abazaba survivor kuri iki cyorezo uko byanyira kose ntabwo kizakurikira gishobora kubashyira hasi, kiramutse kije mu buryo nk'uko iki ngiki kirimo kidusaba kwambara mask, gukora social distance n'ibindi. Niyo mpamvu rero Ubuyapani nabwo bwakomeje kwihagararaho, ariko ubundi naho ibibazo ni nk'ahandi hose. So sinza kweruka kuri structure ya coronavirus cyangwa se coronavirus structure, Dr. Uri yayisobanuye neza cyane, and then Dr. Felix also nawe yayivuzeho. Icyo ngarukaho nuko harimo abantu bagiye bavuga ngo corona ni izina ryazanye na Donald Trump, perezida wa United States, ariko ntabwo ari byo, ni izina risanzweho kubera iyi forme y'iyi virus ifite ijya kugira ikintu kimeze nka corone y'abamikazi cyangwa se bamisi. So ni ibyo rero naho bihera bakayita corona, ariko ubwo ni amazina bayaje gutyo, abantu bakicara bakareba izina rishobora kuba ryaba ribereye. Ikindi nuko muri structure zayo hari igihe ifite ibice byinshi bitandukanye, harimo bagiye bavuga za envelope ya positive sense cyangwa se stranded RNA na za mRNA genome, ariko icy'ingenzi nuko ifite n'icyo bita lipid bilayer. Murebye kuri iyi foto mushobora kubona ikintu kimeze nka structure kisa naho kiri thick ukuntu, ariko niyo mpamvu abantu muri public health expert baduha inama z'uko gukaraba amazi n'isabune bishobora kugabanya kugabanya iyi virusi. Niyo mpamvu kubera ko iyi lipid layer, isabune isanzwe n'amazi ishobora kuyidisruptinga, bikaba byatuma virusi quantity za virusi muri environment zigabanuka. Ikindi nuko nabyo babigarutseho simbitindaho, iyi virusi ubundi cyangwa se iyi SARS-CoV-2 ubundi iri mu kategori y'ama coronavirus, ariko ubundi human coronavirus ntabwo zajyaga ziteza ibibazo byinshi cyane, kuko ubundi zitera 5 to 10% of common cold. Izi ndwara z'ibicurane turwara inyinshi bazitewe n'izo virusi zirimo izo kategori, ariko iyi ngiyo yaje ari yindi. Ikindi nuko ubona ko outbreak zazo wasanga ari nkeya cyane, ku buryo ni za outbreak ntabwo byari byagera ku rwego rwa pandemic nk'uko Dr. Felix yabisobanuye. Icyo nagaruka nuko muri ntuze upper respiratory symptoms zimwe na zimwe tumaze kuzimenyera, harimo croup nicyo twita ngo laryngobronchitis, no gufungana mu mihogo, abantu bakagira dry cough, then abantu bakagira rarely pneumonia n'ibindi. Mu bana ho akenshi izi virusi yo zatatse abana zitera za diare, ibyo ni ibintu dusanzwe tuzi. Iyi novel coronavirus rero niyo itandukanira na zira ziri normal human coronavirus, kuko yo uretse no kuba ifata igice cy'ubuhumekero, igera no hasi kinjira mu bice byo hasi cyane, kuko byari bigoye ko izindi virusi ushobora kuba zagerayo cyangwa se zikaba zahatinda, ariko iyi SARS-CoV-2 yo ifite uwo mwihariko. Niyo mpamvu ubona ko irimo inateza ibibazo cyane, kuko za case fatality nko muri za 2002-2003 ni zagiye ziba ziri hejuru, mu rutateri zari ziri hejuru, ariko nubwo abantu bagerageje kuba bayi-containinga cyangwa se containment in a public health term. I'm very sorry, I'm using really very layman language kugira ngo byibura mugire icyo mutoramo, otherwise ngiye muri deep term ntabwo abantu baza kumva icyo ndi kuvuga. So Dr. Uri yagarutse ko iyi virusi yinjira muri serire y'umuntu, nabyo ntabwo ndibuze kwitindaho. Murebye kuri image iri on the left side, hari uko virusi yinjira ijya kuri serire igahagararaho, ifite uturecepteurs dutuma ibasha guhagararaho, kuko ni nkuko sinzi uko twabigeranya, ariko nuvuga ngo buri virusi cyangwa se buri bakteri, buri kintu cyose gifite ikintu gituma yinjira. Ibifite nka mu Kinyarwanda tugita ngo ni nk'icyitso, niko nakita. Donc hari akantu kaba kari aho ngaho gashobora katuma ikintu kivuye hanze gishobora kuba cyakinjira, birahuza, bigatuma rero ya virusi yinjira muri serire y'umuntu, ikanyura muri procedures and cascade phenomenon nyinshi imbere muri serire kugira ngo izongere gusohoka yamaze ku bindi virusi ishobora kujya hanze ikaba yakanduza undi muntu. Kuri image y'Iburyo murabona uko ibigenza mu bihaha by'abantu. Kuri image ibanza kuri image ifitemo utuimage tubiri, hari before infection and after infection. So actually after infection nuko yimaze kwinjira mu bihaha ituma turatufuka turi mu bihaha twitwa za alveola cyangwa se za alveolus twuzura amazi, bigatuma rero umuntu adashobora kuba yahumeka mu buryo bwisanzuye. Ndabigarukaho hasi muri pathophysiology irambuye. Iyi pathophysiology rero nko tubivuga, ubundi pathophysiology ni uburyo muri medical term indwara ishobora kuzahaza umubiri cyangwa se uko yitwara mu mubiri, kuko hari physiology isanzwe. Physiology ni uburyo buri fonctionnel cyangwa se functional mu buryo buri normal umubiri ukoramo, ariko bugiye mu buryo buri abnormal, that becomes pathologic. So it's pathologic or pathologic physiology. Ni ikintu kitaba cyifuzwa mu yandi magambo. Iyi pathophysiology rero, iyi chart ishobora kuba itari buze kugaragara neza, ariko ndabaha incamake. Hari uburyo abantu bakomeje kujya bakora ubushakashatsi cyane cyane muri transmission n'uburyo virusi yipresenta and so on. Ariko so far ndeberanywa na Dr. Uri and other medics on the platform ko pathophysiology or pathogenesis ya COVID-19 ntabwo iraba elucidated mu buryo buri clear cyane. Harimo duce abantu bagiye bazi, ariko ntabwo biraba byinshi uko tubisanzwe tubizi ku zindi ndwara, cyane ko noneho hagenda hazamo nizo za mutations zitandukanye zituma ibintu bimwe na bimwe bigorana kubyumva. Ariko muri iyo pathophysiology harimo igice cya icyo twita ni asymptomatic phase na symptomatic phase, birumvikana. Muri asymptomatic ni igihe umuntu ashobora kuba afite virusi ariko atagaragaza ibimenyetso inyuma, ariko n'ubundi nk'uko bamwe babizi ushobora kuba ufite virusi utagaragaza ibimenyetso ariko n'ubundi ukaba wakanduza. So muri asymptomatic phase rero, ubundi SARS-CoV-2 yinjira muri respiratory aerosols. Izo aerosols ikabindinga kuri nazo epithelial, agahu kaba kari mu ntuza, envelope kaba kari mu mazuru, then iyo biba bikiri muri upper tract, muri ya nzira yo hejuru y'ubuhumekero. Ariko the main host receptor for viral entry cells ubundi niyo cyo twagarutse cyitwa AC2. Iriya niyo y'ingenzi cyane, nakivuzeho hejuru, ku buryo uretse no kuba zira AC2 receptors zigaragara mu maseri ari hejuru mu nzira z'ubuhumekero, ariko ubundi hari n'izindi organe zigiye zifite. Ni nayo mpamvu Dr. Uri yabigarutseho ko ntabwo COVID-19 izahaza ibice by'ubuhumekero gusa, cyangwa se SARS-2 izahaza ibice by'ubuhumekero gusa, inazahaza n'ibindi bice by'umubiri, cyane cyane bifite hahandi ishobora kwinjirira, kuko imaze kwinjira muri icyo twita ngo respiratory tract, ijya muri circulating system y'amaraso, aho rero ishobora kugera hose, haba mu mutima, mu mpyiko, ishobora kuhafata bitewe n'uko hari favorable mu gifaransa kuri virusi. Iyo imaze kwinjira rero igera cascade, cyangwa se hari ibice byinshi inyuramo, ikireplicatinga, ni ukuvuga ngo igenda ni nko kwimultiplier ikihinduranya niko nabyita mu icyo kinyarwanda kandi ikanakora propagation. So icyo rero iyo propagation rero ubundi nayikora bite nuko ibiri nafashwa n'izo ciliary cells izo twita ngo ni cells ciliary ziri mu bice by'ubuhumekero, and kuri iyo stage usanga imara iminsi mikeya cyane kuko immunity response ubudahangarwa bw'umubiri cyangwa ubwirinzi bw'umubiri baba buri kugerageza kurwana nayo, noneho umuntu akaba yagira viral load iri hasi, ariko nubwo bitabujije ko ashobora kwanduza. Ni ukuvuga ngo viral load bishobora kuba iri hasi, ariko muri case y'iyi SARS-CoV-2 umuntu ashobora kwanduza. Kandi noneho niyo bakoze test ya nasal swab ka kantu bakoze mu mazuru kaga cotton, n'ubundi examen ziza cyangwa se resultat ziza ziri positive. Iyo rero tuvuye muri phase ya asymptomatic, tureba noneho uburyo iriya virusi yinjira cyangwa se yataka ibice byo hasi by'ubuhumekero. Uretse kuba hejuru mu mazuru, iyo igeze hasi bigenda bite? Ubwo ni mu bihaha. Kuri iyi stage rero habaho icyo bita migration ya virusi iva muri nasal epithelium. Nasal epithelium ni za membrane ziba ziri hejuru mu mazuru hejuru, ziba zitwikira amasere agize amazuru hejuru, and then ikamanuka muri airways, donc ikamanuka muri za nzira z'ubuhumekero zisanzwe mu mihogo, ziciye hirya ngiyo mu nkanka zikamanuka hasi. And ahangaha cyane cyane baravuga ngo due to the involvement of the upper airways, disease manifest with symptom of fever, malaise, dry cough. Urumva umuntu agira umuriro, akagira ibintu byo kumva yacitse intege, akaba yakorora. Then aho haba ari icyo bita immune response during this phase, izanamo ibindi bintu bita chemocytokines. Ni abantu bashobora kubyumva neza, ubwo sinzi niba ndibuze kwisobanura, ariko ubundi ni cytokines, ni cellules zishobora gufasha muri inflammation mu bwirinzi bw'umubiri kwa. Ndimo ndageza kureba ko nabyumvikanisha. And then izi ntuze za chemokine ligands cyangwa se chemokines interferons ziba ziturutse kuri za cellules ziba zabaye infected, kuko cellules zibaye infected zitaziririzinga utundi turemangingo cyangwa se utundi duce dushobora gutuma haza kuboneka uburyo abasirikare b'umubiri bagomba kumenya ko hari icyateye wa mubiri, bakaza kureba uko barwanya za virusi. So aha rero nuvuga ngo majority y'abarwayi akenshi ntabwo barenga iyi etape, usanga rimwe na rimwe umubiri wirwanyeho ukaba wazirwanya, then bakaba bakira nta n'ikibazo kibayeho, ariko still bashobora guspreadinga infection. So akandi gafazi gakurikiramo nuko muri iyo lower respiratory tract, nuko noneho bimera bite kugira ngo umuntu yamaze kurwara, amaze kwandura virusi, abe yagira noneho yatwita ngo ni acute distress syndrome, kugira ngo noneho guhumeka bibure. So about 1/5 of all infected patients progress to this stage of disease, and they develop severe symptoms. Nuvuga ngo kimwe cya gatanu cy'abarwayi abenshi bajya muri iyo stage, binjira muri icyo cyiciro, and virusi noneho yinjira muri alveolar epithelia, iba yinjiye noneho yatangiye kugera mu bihaha, and then iciye muri ya S2 twavugaga, kuko ira niyo key, kuko iyi S2 urebye iri very common mu masere ya respiratory tract cells z'abantu bakuru. Ari nayo mpamvu ntekereza ko ubanza wenda maybe Dr. Uri yigeze abivugaho, abana haracyarimo amahirwe ko bashobora kuba badashobora gufatwa n'iyi virusi. Hashobora kuba ari impamvu abantu bashobora kuzagenda highlightinga later. So virusi yinjira mu masere y'ibihaha cyangwa se y'ubuhumekero, and then igatuma noneho umubiri uririzinga izo naho zita cytokines and inflammatory markers. Inflammatory markers ni uduserire tuba tuza cyangwa turemangingo tuza kugira ngo dukreye ubwirinzi bw'umubiri. Akenshi muzabirebe yukomeretse ku ruhu hahita hatukura. Kuki hatukura? Burya imbere habayemo cascade y'ibintu byinshi bishobora gutuma umubiri ubasha kurwanya cya kintu gitumye niba ari nk'akasimba kakuriye cyangwa se ari nk'akantu kakunoshe, umubiri uhita ukanguka ugahita wohereza abasirikare. Aho rero bazi ni nko kuhabyinagira, niko nabyita mu Kinyarwanda. Baraza bakahabyinagira kugira ngo babashe kurwanya cya kintu kihateye. Uko kuhabyinagira rero urumva kuhasiga ibikomere. N'izo inflammation rero nicyo kimwe, ni system mu mubiri ugira biri normal mu kwirwana, ariko ibikurikirizo iyo inflammation nibyo akenshi binatera ikibazo, aribyo tugiye kubona mu gace ko hasi. So izi cytokines rero zikora icyo bita storm ya cytokines, za serine ngo mbabwira zikora storm ya cytokines, and then na za neutrophile n'amaserire aturuka mu mubiri aba ashinzwe kurwanya virusi zinjiye mu mubiri cyangwa se utundi dukoko twinjiye mu mubiri. Hakaza za CD4 zo ndumva muzizi cyane cyane muri HIV, abayifasha helpers ba CD4 cyangwa se CD4 helper, na za T cells. Ni amaserire menshi afite kategori nyinshi zitandukanye, kandi byose icyo ifite ikigenda ikora, noneho zigatangira kuba nyinshi mu bihaha, kuko urumva twageze hasi mu bihaha. Noneho rero icyo ya cascade, kuko izo cells ziba ziri responsible for fighting off the virus, but in doing so they are responsible for the subsequent inflammation and lung injury. Nibyo nababwiye ko ni nk'aho abasirikare barwaniye, uko byagenda kose ntabwo habura icyatsi cyahakomerekera cyangwa se cyahasigara, cyangwa se inzovu wambwa ariko cyatuvuga mu Kinyarwanda. Aho rero ni naho ruzingiye, kuko izo inflammation zasigara nizo zituma ibihaha bigira ibibazo. So the host cells undergoes apoptosis with the release of new viral particles. Apoptosis ubundi ni ugupfa kw'amaserire. Amaserire menshi arapfa, arangirika, and then virusi ikabasha no gusohoka, kubera ko iba yinjiye mu maserire imbere, ibonye umwanya wo gusohoka ikajya noneho hanze, and igatangira no ku-infecta izindi za alveolar zari zirya zari zanabaye infected muri ya epithelial cell tugiye tugarukaho kare. And due to the persistent injury caused by those sequestered inflammation, inflammatory cells, and viral replication leading to loss of both type 1 and type 2 pneumocytes, hari icyo bita diffuse alveolar damage, and then eventually bikatuma umuntu ananirwa guhumeka. Niyo ya foto nigeze mbereka hano haruguru kare. Aya mazi ntabwo bayazanyemo n'ikindi, ni mechanisme y'umubiri iba yabaye, then za alveolar zikuzura zo za inflammation. Amazi aba yaje ni nko kuvuga baje nko kuzimya umuriro, niko nabyita. Ndibona ko Ikinyarwanda kiri very vulgaire mu Gifaransa, ariko no kugira ngo bashe kumva neza in the simple way uko mechanisme zigenda. Iyo rero cascade nari ngarutseho niyo ituma ibihaha byangirika, cyane cyane bihereye muri za alveolar, twadufuka dutoya umwuka winjiramo. Aho rero niho ruba ruzingiye, nibwo umuntu atangira kubura uko ahumeka, noneho tukajya mu buryo bya bimenyetso bigaragara, umuntu akabura umwuka, gukorora birumvikana, kubera ko hejuru habayemo twa tuntu twinshi tutuma ntuze tract ifungana, akaba yakorora. Mu bana ho hari na virusi zinatera n'amadiyari mu bana, ariko muri case ya covid ubwo tubisize ku ruhande. Ariko muri make za symptome abantu bazi, hari ukurwara umutwe, hari ukugira ute, noneho bikahurirana ugasanga umuntu ararembye. Hari umwarimu wigeze atanga ikiganiro mu minsi yashize kuri television ya hano, avuga ko ubundi nature uko ikoze ntabwo twagombye kuba tugirana ibibazo na nature. Nuvuga ngo virus ntabwo twagombye kuba tugirana ibibazo nazo, ariko birashoboka ko iyo zinjiye mu mubiri wacu, umubiri wacu ahubwo ari ushaka kwirwanaho, akaba ari uduteza ibibazo, bigatuma rero none za virusi nazo zirwanaho. Ni nko kuba umuntu yibereye aho ngaho, wagira nk'amahirwe makeya uri nko mu nzu ukabona nk'ingwe irakwinjiranye, ugatangira kuyirwanya, maybe ashobora kuba yanakwihorera, ariko watangira kuyirwanya igahita ikwereka aho ibera akaga. So uwo mu professor yagaragaza ko abantu bakwiye kureba ko dushobora kubaho muri environment in harmony, kugira ngo turebe ko twakwirinda ibyorezo bimwe na bimwe. Gusa in some cases ntabwo byoroshye, kubera ko nka virusi ntabwo wabona uko ubana nayo, kubera ko ikwinjira utabishaka, uko byagira kose habaho kwangirika. So muri symptome none nazigarutseho, hari zigaragara mu cyumweru cya mbere, hari zigaragara mu cyumweru cya kabiri, ntabwo ndibuze kubigarukaho cyane. icyo nagaruka nuko mu cyumweru cya kabiri cya gatatu abantu benshi usanga babaye hospitalized, cyane cyane kimwe cya gatatu cy'abarwayi bakenera za ICU care, nibwo mubona bakora za intubation, and then hakabaho rimwe na rimwe ko twavuga za cytokine cyangwa storm zituma habaho na multi organ failure, kubera ko umubiri urakorana, iyo ibihaha bitameze neza, amaraso ntabwo agenda neza, ntabwo abona oxygen, ubwo brain irahangirikira. Harimo na za hypothesis ko na virus ishobora kwizamuka yajya no muri brain. Those are the discussion under table. I'm not clear about it, and I don't really want to discuss those kind of things. And then muri late stage hazaho noneho na za sudden cardiomyopathy, umutima nawo ugafatwa, and then viral myocarditis and later cardiac shock. Nibwo rero umuntu ubona yitabye Imana kubera izo mpamvu. So ni impamvu rero zuko yinjiriye mu mazuru, imanuka hasi mu muhogo cyangwa se mu buhumekero, ijya mu bihaha, ibyo ikora mu bihaha mu bisobanukiyeho gakeya, and then n'impamvu umuntu azahara iyo yagiye muri iriya fazi. Ndumva rero nicyo nabasharinga uyu munsi as a pathophysiology. Nibaza ko buri wese that term of pathophysiology yayumvise, kugira ngo mu gihe mwandika inkuru, kuko most of you muri abanyamakuru, mu gihe mwandika inkuru mushobora kujya mucishamo mukareba n'izo science behind, mukaba munumva neza what is really in, kugira ngo hatagira ushobora kuba yakandika false information cyangwa se ibintu biri baseless bidafite evidence, kandi already hari evidence bazihari, scientists, researchers baba baragiye bakoraho. Iyi slide ya nyuma rero ni bwa buryo busanzwe, hari uburyo virusi zanduriramo kugeza ku munota SARS-CoV-2 cyangwa se COVID-19 tuyibona biciye mu mwuka cyangwa se mu matembabuzi. So cyane cyane niyo mpamvu banavuga ngo dushyiramo social distancing, kuko ushobora gushyiramo social distancing bigatuma cyangwa umuntu ashobora kuba yemeta ari kuvuga ashobora kugabanuka ntakugereho, nubwo rimwe na rimwe iyo bibaye aerosol biba bigoye kuzirinda, ariko mask nicyo nayo imaze ikagabanya the quantity of virus we can inhale, and then maybe there is a chance ko twagabanya morbidity and mortality. Hari nabwo uburyo bwo gukaraba intoki, hand wash technique, nabwo ni ukubugarukaho mukajya mukomeza kubwigisha abanyarwanda mu buryo bumwe cyangwa se ubundi. Ndibaza rero ko ntagiye muri byinshi biri too medical and too complicated terms. Ni muri make n'ibyo nashaka kubasharinga kuri pathophysiology ya COVID-19. Nizera ko maybe there is something you have picked, and then if you have any question, any inputs, please just go into chat box, and then we can respond as much as we can. And already we are with other experts in the panel, so maybe they can also support any comments or question if there is any asked to me. Murakoze rero, bifurije ibihe byiza.
Murakoze cyane Dr. Singa. Thank you so much for your presentation. It was well explained and we have really learned a lot. I would also like to thank everyone for the participation. Yeah, thank you so much. There are so many questions in the chat box. In order for us to give Dr. Singa time to breathe, I will welcome Dr. Daisy to answer the questions related to pregnant mothers and COVID-19 vaccines related. So welcome, Dr. Daisy. We'll use a few minutes. You will explain to us about that. Now we need to go to so
mwiriwe, nitwa Daisy Umutoni, ndi umuganga, nkaba nanjye mfite PhD muri biomedical research. Mwakoze kwitabira kandi mwakoze no kubaza ibibazo kugira ngo mubyumve kurushaho. Okay, nari nabisubije muri chat, ariko wenda reka nongere mbivuge. well, iby'abagore batwite ngira ngo yabajije, hari umuntu wabajije impamvu badakingirwa, kandi Dr. Felix icyo yagishubije. Mu bihugu biterwa n'igihugu, ibihugu bifite amabwiriza atandukanye, nko muri Amerika n'ahandi barabemerera, baravuga ngo kereka babanze bavugane n'abaganga babakurikirana barababyaza, kereka iyo afite nk'ikibazo kiri special kuri we atari ukuvuga ngo ni uko atwite, uwo muganga wenda akaba yamubwira ati uretse gufata urukingo, ariko ntago babuza gukingirwa. Mu Rwanda ho bashyiramo caution, hari information z'uko batabemerera. So as Dr. Felix yari yabivuze, bishobora kuba biterwa wenda nuko babona ubushobozi bwo kubafasha baramutse bagize ikibazo budahari nk'ibihugu biteye imbere, ariko ntago ndishuwa impamvu wenda leta itemerera abagore batwite mu Rwanda gukingirwa, ariko nta bushakashatsi bakoze muri specific groups. Tuvuge hari uwabajije ko kubera arwaye kanseri, hari well, sinzi niba ari we cyangwa yabarizaga undi, hari abagore batwite, hari special cases nk'izo, conditions zitandukanye. Unfortunately ntago ubushakashatsi barakora research mu ma group ari specific. Tuvuga abana bato cyangwa well, murabizi ko abana bato batemerewe gukingirwa below 12 years, abagore batwite, n'ama condition atandukanye bazagenda babikora with time, bakora ama research ari specific muri izo groups, bagende batanga recommendations. Hari uwabajije ukuntu wakwirinda utwite, ni nk'uko undi muntu wese yirinda, ntago hari specific recommendations. Ni gukaraba intoki, kwambara maske igihe uri hanze, gushyira intera hagati yawe n'abandi. So gusa nyine kubera ko umugore utwite aba ari more at risk yuko arwaye yaremba, bisaba ko arushaho no kwirinda kurusha abandi nyine, ariko ingamba ni za zindi. Naho uwabajije ikibazo cya ngo ashobora konsa, ashobora gufata urukingo arimo konsa, again nawe ni ya group nanone batarakoraho ubushakashatsi buri specific, ariko so far ntago bibujijwe. Ntacyo urukingo rwagira ku mwana, ahubwo hari ubushakashatsi bwerekana ko iyo akingiwe, abasirikare bakora nyine urukingo rukora rutuma umubiri w'ubyara, ubudahangarwa umubiri w'ubyara kubera urukingo yahawe, nabwo bushobora kunyura mu mashereka, ahubwo bugaprotectinga n'umwana ari konsa. So nta kintu kibujijwe specifically cyamubuza gukingirwa, kereka afite ikindi kibazo runaka, nabwo akavugana n'umuganga we, ariko kuba yonsa ntago byamubuza gukingirwa. So murakoze, nizere ko nashubije, ariko na bagenzi banjye hari icyo nibagiwe banyunganira. Murakoze.
Thank you so much, Dr. Daisy. I welcome Dr. Uri. Nawe aduhe ibisubizo ku bijyanye na the questions related to vaccines, to cancer patients, and things of the sort. So, Dr. Uri, you're welcome.
Okay, thank you, Madam Moderator. I was actually addressing these questions relating to cancer patients on the chat. So there were a couple of questions posed by a participant here, Mr. Gasirikare. Let me try and find these questions. Okay, so the first question was, you know, if I'm a cancer patient, should I get the COVID-19 vaccine? Okay, and then the second question was, as a cancer patient, if I get sick from COVID, if I get infected and get sick of COVID-19, how does that affect my cancer prognosis? Okay, so my answer to that is that, to the first question, as to whether a cancer patient can get the vaccine, I think it depends on, and this can be best answered by a personal physician to the cancer patient. But my answer to that would be, if the patient or the patients are on chemo or radiotherapy, which severely immunocompromises the patient. So the chemo and the radiotherapy substantially reduces and weakens immune function. My educated guess would be that a patient on chemo or radiotherapy is not best advised to have the vaccination then, until the patient gets off the therapy, because the vaccination is intended to elicit an immune response in a person, and the immune response cannot be elicited if immune function is already weakened with chemo or radiotherapy. So the cancer patient would have to complete the therapy, eventually have a restored immune function, so that the person can become vaccinated. Then there's the second question of, okay, if one as a cancer patient gets COVID-19, if one gets infected and develops COVID-19, does that affect the cancer prognosis? I was actually trying to answer the question on the chat here. I haven't posted it yet, but I was going to say that you know, having cancer, first off, increases your risk of developing severe COVID illness in case of an infection. Okay, and then infection with the virus, particularly with these circulating variant forms of COVID-2, does not help if you're a cancer patient. So in terms of prognosis, I think, but I don't have any concrete evidence for that, for what I'm about to say. But I think infection with COVID-2, particularly with the variant forms, if that increases, if the cancer itself increases the chance of developing severe illness, it doesn't help with the prognosis. So those are my answers, or those are my responses to the issue of addressing the question of cancer patients either being vaccinated or in the event that they develop COVID-19.
Thank you so much, Dr. Uri. There are so many questions in the chat box, and because of time, we cannot be able to exhaust all of them. But we have a session tomorrow and the day after that. What we can do, I think I will welcome Dr. Simba to give us closing remarks and answer a few questions that were asked about vaccines. If you can take all 3 vaccines, yes, yeah. So welcome, Dr. Simba, and then after Dr. Simba, we can close. Thank you so much.
Yeah, thank you so much, moderator. And also thank you so much for everyone who has really managed to attend today's sessions. Actually, this is a very informative and also it's a good initiative that brings on journalists, and then it's really desirable that once you are writing, you really are conversant with something really tangible about this COVID, and then even the general population can really get something in terms of increasing the awareness and knowledge about the disease. So I'm not sure if I can really respond those questions, because some of them, they are out of scope of our expertise. But let me say that ku bijyanye na za vaccine and pregnancy and how many doses we can get, of course Dr. Daisy yabivuzeho. Umudamu utwite aba ari kuri risk y'ibintu byinshi, harimo advantage, harimo na disadvantage zo gutwita, ku buryo kuvuga ngo twemeze ko agomba kuba na vaccine, it's mandatory, it's still early. Hagomba kuboneka evidence, ubushakashatsi ko it's safe, and then once it's okay, then that will be all right. Ariko to the other extent, umuphysician cyangwa se gynecologist or obstetrician ukurikirana umudamu utwite aba afite all how can I say alternative mu biganza bye byo ku assessing the situation, and nakaba recommending what to do based on maybe the knowledge, the education, as Dr. Uri said, the common knowledge of an educated person, akaba yareba niba hari kitabangamira ubuzima bw'umubyeyi. Ariko when you are really pregnant, you need to make sure that you take all precautions measures mu buryo bwose bushoboka, haba gukingirwa, haba kwirinda. Kwirinda byo ni obvious, ni no kwirinda kurusha n'abandi bose, kubera ko umugore utwite aba afite undi muntu mu nda ye. Urumva rero aririnda akanarinda na wa wundi atwite, kuko hari theory zakumye kujya zisirikira ko ibintu bimwe na bimwe ntabwo bishobora gutraversa the placenta barrier ngo bijye ku mwana. Ariko so far there are some new hints that maybe some of the harms can also cross that barrier zikaba zaricinga umwana. So ni ibintu n'abantu bagomba kwitondera mu gihe science irimo yidevelopa, kugira ngo abantu bazabe bafite amakuru meza ahagije wenda ejo hazaza. Ariko mu gihe abantu bakiri kurwana nabyo, we need really to pay attention and follow the instruction of the scientists and also doctors as well as pharmacists, kubera ko na pharmacy buri hari ubumenyi bafite ku bintu by'imiti n'inkingo. So lastly, for the doses, so far WHO cyangwa se n'abandi ba providing izi nkingo are recommending 2 doses. So the first dose, and then you put in a window of 20 something days, and then or 14 days, I think 14 or 21 days, and then you get a second dose. Ariko already query variant ya Delta, which is actually very virulent, if I can say like that. It is really very virulent, ikaze kurusha. Nibaza ko harimo uburyo abantu harimo batekereza niba if people can get the third dose. So those are the discussion on the way. Actually, I was reading some journals that in the UK, maybe they are now actually thinking how fast they can be to get that third dose, which they call the booster. Ni nk'icyo kongera noneho immunity nkeya rimaze kuzamuka kuri za dose ebyiri, noneho ukayiha ikintu kimeze nka pisto ikazamuka cyane, kugira ngo ina covering na za Delta variant zirimo ziza n'izindi variant zishobora kuba zaza ejo cyangwa se ejo bundi. So icyo rero navuga ni uko science up to now iracyari very ntabwo navuga ngo iri confused, iracyari unclear for certain things about COVID-19. Hari very good steps zamaze gufatwa muri research, nk'aba Dr. Uri barabizi, they are working on those kind of things. Ariko hari steps zamaze gukorwa mu kumenya neza what is this virus. Ariko hari n'ibikiri byinshi bikiri inyuma bitaramenyekana neza, kuko up to now ibyo dukora byose ni preliminary advice zabonywe COVID kiza, n'izindi zigenda zigaragara uko iminsi igenda yicuma. Ariko ntekereza ko ntabwo bihagije kugira ngo tuvuge ngo this disease we can really deal with it. Let's give an example of malaria. Malaria turabizi neza ko yandura n'ukubera mosquito. Umubu utakuriye ntabwo wakandura malaria. Ntabwo umuntu yakuruma ngo nakuruma cyangwa se ikindi kintu kiguteme ngo kikwanduze malaria. Umubu wonyine niwe ushobora kukwanduza malaria. Kandi buriya n'imibu yose nayo si kimwe, hari imibu itanduza malaria. Ni ukuvuga ngo rero birazwi neza uko malaria yandura. Wenda muri no minsi naho hatangiye kuboneka ubundi buryo bushyashya malaria ishobora kwanduramo, nuko uburyo bushyashya imibu ishobora kurumana, kuko twatumenyere ko irumana nijoro, ariko ubu hari uburyo buvuga ngo ishobora no kuba inarumana ku manywa cyangwa se mu gihe hakiri kare mu masaa 12. So ariko byibura hari science iri very clear about malaria, nubwo abantu bagikora ubushakashatsi kuri byinshi kuri yo. Ariko compared to COVID-19 or SARS-CoV-2 as a virus, biracyari kare ko abantu bashobora kugira ibyo bahagarara bavuga ngo ibi ngibi ndabyemeza ko ari uko bimeze. Ibintu byose ni ukurikiza amabwiriza ari current duhabwa n'inzego z'ubuzima, inzego z'ubuvuzi, kugira ngo turebe ko abantu bakomeza kunguka ubumenyi bwatuma icyorezo tukihashya burundu. So mu kurangiza rero ndibaza ko nashimira buri wese witabiriye. I'm not actually, maybe I will not be the one to close, but maybe just giving a summary or closing summary. Nashimira bitabiriye, twabonye ibiganiro byiza, hari kuduha current status twahawe na Dr. Uri. He's an expert muri research. No, he's a medical scientist. No, maybe he's not a medical doctor, ariko ni medical scientist, ariko ntekereza ko bya nabyo it's really an added value, kuko hari ibyo abazi nkatwe babaganga tutaba tunazi. So birumvikana rero ko ni ukuzuzanya. Ikiganiro taze rwose, it's very well elaborated, and you will get the notes online, and you can read later and listen to this recorded session, which is also, it's another chance to understand more, and you can even ask more questions. Twataza na contact zacu. Dr. Felix, it's the same, yaduhaye background. Hari nk'abavuga ati byagombye kuba byabanje tukakurikizaho ibindi nyuma, ariko it's okay as long as you understand that yagombye kuba byabanje. Nuko wanumvise neza ko nyine it was obvious ko gishobora kuba yacyagombaga kubanza, ubwo muri make wanabashe kubitandukanya. Icyo natanze ni cya pathophysiology, which is actually a little bit complicated, kubera ko bituma ninjira muri medical science cyane, ariko we try to be as simple as possible kugira ngo mugire icyo mukuramo. Ndibaza ko rero that's a good chance, that's a good opportunity for all of us, you journalists and also we as scientists, to exchange and also try to see what we can do for our population, what we can provide for our citizens, our fellow Rwandese, so that they are enlightened more about this disease. And mugihe tuvuga ngo ni mugiye muri kuba mu rugo cyangwa se mu gukore social distancing, bakaba bumva impamvu tubabwira, atari uko hari ukubahima cyangwa kwicisha inzara cyangwa se ibindi, no kugira ngo turengere ubuzima bwabo, kuko haguma amagara, burya ibindi baravuga ngo amagara, haguma amagara ngo ibindi barahaha. So nuvuga ngo umuntu ari muzima ashobora no kuba yakora ikindi gishobora kuba cyatuma ejo bundi akomeza kuramba n'igihugu kigatera imbere. Ndumva ari ibyo navuga. Hasigaye we are available. I shared my contacts if anyone wants to contact me for any topic related to medical, global health, so feel free to contact me. Thank you so much. I hand over to you, moderator.
Thank you so much, Dr. Simba. The questions are still coming in, but we are out of time. We have another session tomorrow, and we can continue with the questions. I'd like to thank all the presenters, doctors that have been able to teach us all these things that they have. I also want to thank the participants for participating, the interactions and everything. I hope to take the good news and the right things that we have learned here as we write the news. I would like to welcome Mr. Rwandan-EAC from Health-Edu to give us the closing remarks on the program for tomorrow. Thank you so much. You're welcome, Mr. Rwandan-EAC.
Yeah, thank you very much. Let me give, let's give 1 minute to Dr. Felix, and then I can come later. Maybe 1 minute for Dr. Felix.
All right, thank you. You're welcome, Dr. Felix.
Thank you so much for this session. Ikintu kimwe nshaka kuvuga ngendeye ku bisubizo bimwe nanone bagiye batanga ku bibazo mwabajije. Kenshi twagiye tuvuga ngo umuganga wawe buriya ni umuntu ukomeye cyane, kuko aba afite amakuru undi muganga rimwe na rimwe adafite. Ngo muganga wawe aba afite amakuru undi muganga aba adafite. Niyo mpamvu buriya iyo bishobotse kumugisha inama aba ari byiza kurenza uko wajya kuzigisha undi wese. Nk'ibi ngibi ndabigarukaho wenda ngendeye ku kibazo uri yaje gusubiza, kuko nanjye nanakivuzeho kuri cancer patient bakeneye gufata vaccine cyangwa se ntibazifate. Ikibazo nyamukuru gihari, kuko nanjye ngisubiza nanigeze kugisubiza muri chat, nari navuze ngo your treating medical doctor aramutse akubwiye ko ufata vaccine wayifata. Kubera iki? No kuvuga ngo aho ngana bivuze kubera ko umurwayi wa kanseri, ikintu cya mbere ndabanza kuvuga, kanseri twebwe iyo tuyumvise tuyifata nka malaria. Nanone kanseri itandukanye nuko dufata malaria. Buriya ngewe iyo uje ukambwira ngo umuntu arwaye kanseri, okay, ntimuze kubyumva nabi, nge no kuba nakoze specialization muri diagnosis of cancer, niyo specialty yanjye. Iyo rero umbwiye ngo umuntu arwaye kanseri, nge ntabwo umpaye amakuru afatika, kuko umuntu urwaye kanseri y'amaraso atandukanye n'umuntu urwaye kanseri y'ibihaha, atandukanye n'umuntu urwaye kanseri yo mu bwonko, atandukanye n'umuntu urwaye kanseri y'ibere. So niyo mpamvu rero haravuze ngo muganga wawe ni uzi niba ushobora gufata vaccine cyangwa se utafata vaccine. Muri rusange kanseri zose zizatera immunocompromission, zizagabanya immunity, ariko nanone mu gihe bari kukuvura, no umuganga uri kukuvura azi phase ugezemo. Azi niba uri muri phase aho immunity yawe iri hasi cyane, azi niba ugeze muri phase aho immunity yawe iri kuba restored, azi niba uri muri phase aho immunity yawe iri okay, ushobora kubona vaccine. Niyo mpamvu rero igihe ufite icyo kibazo, uwo muganga ukuvura niwe ufite amakuru yose, niwe uraguha igisubizo kirimo wa accurate. Kuko niba uri immunocompromised nkuko Dr. Uri yabivuze, ntabwo ugomba gufata vaccine. Ariko niba immunity yawe yamaze kuba restored, uri muri ibyo bita remission phase, muganga wakabona ko uri muri approximately good condition, icyo gihe wafata vaccine. Ibyo birajyana na nyine na wa mudamu twahoze tuvuga. Umudamu utwite, wenda niba twite inda ya mbere, niba twite ya kabiri cyangwa se ya gatatu, byanga bikunze umuganga wamukurikiranye azi ibindi bibazo yagize mu kwezi gushize, mu mezi abiri ashize, ubundi burwayi yagize. Uwo muganga rero gukurikirana n'ushobora gufata decision akavuga ati nkurikije amateka ya naya, nkurikije uko umwana ameze, na Dr. Kariope n'undi yari yabivuzeho, ushobora gufata vaccine cyangwa se ntuyifate. Kandi nawe afata ibyo byemezo nanone agendeye ku mategeko igihugu cyashyizeho. Nkuko nahoze mbivuga, niba igihugu cyawe kivuga ngo ntitwemera vaccine, muri Amerika bayemera, ntabwo uzaba vaccinated. Uyu munsi ntabwo ushobora kujyana iriya vaccine ikoreshwa mu Burusiya ngo uyijyane muri Amerika, kuko Amerika itarayemera, ariko Abarusiya barayikoresha, ibindi bihugu bidafite ubushobozi birayikoresha. So ngewe umwanzuro natanga nako kantu ko kugirira icyizere muganga wawe, akaba ari we uguha advice irenze izindi. Kubera iki? Kuko muganga wawe afite more information, afite byinshi akuziho kurenza undi muganga wajya kubaza. Ubu umbajije uyu munsi aka kanya ntabwo nzi amateka yawe, ntabwo nzi uburwayi bwawe, ntabwo nzi imiti yindi mu minsi yashize wafashe, ushobora kuba we waranafashe akaba hashize ibyumweru 2, ariko uyu munsi ikaba igishobora kuba igifite effect mu mubiri wawe. So niyo mpamvu ngewe as a conclusion, rimwe na rimwe ku bijyanye no kuvuga ngo ese ibijyanye no gufata urukingo, kwivuza kugenda gutya na gutya, better that you consult your treating doctor akaguha more accurate information, kuko afite more information about your health kurenza undi muntu wundi wo ku ruhande. Ndabashimiye, ndizera ko muzakomeza no kwitabira n'izindi session zizakurikiraho kuko nazo zifite ibindi zizatwungura. Murakoze.
Thank you very much, Dr. Felix. Let me take this opportunity to thank all the presenters and the attendees for the time and for the good time that we spent here. Today we had 3 sessions, and tomorrow we'll have 4 sessions, and we shall be very brief and we shall cover all of them. We're going to cover the diagnosis and medical treatment of COVID by Dr. Theonest. We shall cover the physiotherapy management of COVID-19 by Dr. Leila. We shall see how the contact tracing is being done, and in this time by Mugabe Christian, and we shall see the role of ICT in the management of COVID-19 by Alex. So I think it was a pleasure to be with you all, and thanks to the moderator for this period. So we meet again tomorrow at 2 p.m., same link. And we are happy that you are really interested and you are joining and you're asking questions. Thank you very much. See you tomorrow.